Hello, everyone. I'm Dr. Carmen Puliafito, and I'm pleased to have here today Dr. Rajesh Rao from the University of Michigan to talk about stem cells for the treatment of atrophic macular degeneration. Raj, welcome to Retina Synthesis.
Thank you, Carmen. Pleasure to be here.
You've been working on stem cells since you were an undergraduate at the University of Wisconsin and moved on to Yale Medical School and the Mass Eye and Ear. Tell us about your history with stem cells.
Yeah, I was born and raised in Wisconsin, grew up, and then went to undergrad at the University of Wisconsin. And in the late 90s, the buzz all over campus was, this PI, Jamie Thompson, was the first to isolate, derive, expand, grow human pluripotent stem cells. And I didn't, as an undergrad, didn't exactly know what that meant, but I was interested in research and medicine. And the way they described it at that time was, well, here's a chance to study cells that you could never really get access to. Because, in postmortem tissue, there are a lot of cells, even if you can isolate them, don't really regenerate, don't divide, they don't grow. But here was a way really to grow all the important cells uh, that play the key role in all our diseases, such as ALS, motor neurons, Parkinson's disease, dopaminergic neurons. Um, there wasn't as much about the eye at that point, heart attack, cardiomyocytes, but all these cells which do a poor job at regeneration or don't regenerate, we now had a chance to study them. And so even back in the 90s, it was sort of like, well, now there's, a potential for direct cell transplantation. But other areas which haven't been as much leveraged and talked about at that time though was the ability to use these cells for screening for new drugs, and then the kind of science role to model certain diseases of regeneration, and also to better understand human development. A lot of the earliest stages of human development haven't been well studied, and fertility and all that questions then and today. There are high rates of miscarriages and stuff. So as was described back then, this was a tool and a platform to try to unlock our understanding of biology, how to model disease, and also how to potentially provide treatments for diseases where we lack the regeneration, largely in the brain, and talked also a little bit in those early days about the retina. So when I was a freshman there, I was really excited, and I thought, this might be a foray into medicine. I was thinking medical school, but also about innovation and trying to maybe discover something new. So I joined a lab. His name was Suchun Zhang, Ian Duncan. They were both at the University of Wisconsin, who later became major leaders in pluripotent stem cell biology. They were colleagues of Jamie Thompson at the time, kind of considered the pioneer. And I still remember the day, one of the first days I was going into lab, where you stagger the growth of these cells. So one day you may be starting to culture these cells, and then in your cell culture incubator, you may have cells that have been growing for a month or 2 or 6 months. And Suchen Zhang, my mentor at that time, showed me. He was like, okay, here are the cells that I'm starting to grow. And they were kind of a bland collection of colonies of cells. He put the plate back in, and then he took another, you know, plate of cells out, and he said, "Look here under the microscope." And it was just like beating, going back and forth, like coming closer to me in the microscope at 10x, 20x, and what they were, early cardiomyocytes growing. His interests at that time were oligodendrocytes for multiple sclerosis, also motor neurons for ALS. And then he'd show me at different phases of when, this is like 1998 now, different phases of these cells and how to grow the brain in the dish. You look for these phases like neural rosettes, so little rosette structures on a 2D surface. And then he would show me like cells that were telling us that you made a mistake, that you had a contamination, not like a bacteria or fungal contamination, but really you were growing the wrong cells when you intended to grow brain cells. And then so he pulled out another plate of cells which were full of pigment. They were brown, they were black, they were dark. And he's like, these are cells we really don't want on our way to grow motor neurons or oligodendrocytes. And so when we see this, this is a sign of overgrowth, and these are pigmented cells. And I asked, what are they? I was a freshman. I didn't really know much about the eye, but he's like, well, these are, pigment cells that are likely related to the eye, but that isn't really our core interest in my lab. So to make a long story short, the 3 years I was in that lab, we looked at ways of how to grow oligodendrocytes, the myelinating cells, in a more efficient way from these cells. And that was my kind of undergrad thesis. But then, a few years later, I don't know, maybe 5 or 6 years later, um, there were two people there, Su-Chun Zhang, who now became kind of full professor, pioneer in neural differentiation of pluripotent stem cells, and another guy, Clive Svensson, who was also Wisconsin, growing from embryonic tissue neural stem cells. And there was a new faculty member who had just finished his training. He was an MD PhD at Michigan, named David Gam. And he was looking to start a lab at the University of Wisconsin, probably in the early mid 2000s, with both Clive Svensson, now at Cedars-Sinai, comes full circle, I'll explain later, with our current trial, and Su-Chun Zhang kind of saying, hey, here are cells that we're not that interested in as much. We haven't figured out all the tricks to grow them, but they are clearly early eye cells. And I really think David Gam took that and ran with it, because at that time, no one was growing really at that scale, RPE cells. But that's really how I got into it at the University of Wisconsin. And then at Yale, I helped start the Yale Stem Cell Center. The Yale Stem Cell Interest Group in the early 2000s, played a role in state funding for Connecticut for stem cell research, kind of on the coattails of recent CIRM funding, California Institute for Regenerative Medicine, back in the early 2000s. I got involved a little bit with JDRF, who helped the effort in Hartford, Connecticut, to bring state funding for, or to advocate for stem cells. And then during my time at Yale, I did a 2-year, it was called a Howard Hughes Medical Institute Cloister program, where I worked with the NIH Stem Cell Unit, a guy named Ron Mackay, who was at the NINDS, National Institute for Neurological Disease and Stroke, and continued oligodendrocyte research in identifying key pathways in mTOR, PDGF, how to grow these cells more efficiently. Then started my residency at Mass Eye and Ear, where I know you were too, and attached to Mass Eye and Ear as Mass General, and at the time Scapins as well. And then kind of an early question in stem cells is, how do cells make the order that they make? For example, in the brain, you always get neurons before glia. And in the retina, you get ganglion cells and cone precursors before rods and other cells. So I thought epigenetics was a way to better understand that. I learned some of that at the NIH, but these are these patterns of DNA and histone methylation. And you'll find, when you have these DNA methylation, histone methylation, certain kinds of it, that silences that program. So if you look at the glial program, oligodendrocytes and astrocytes, in stem cells, they're defining the order of what they make based partially on the patterns of methylation. And so no one had yet looked at that in the retina at that point. This is 2008 or 9. So at Scapins, I was able to get one of the PIs, Dongfen Chen, interested in, well, when the retina develops, this is in a mouse, in the developing mouse retina, what are the key methylation marks in the retina? So these what define what turns genes on and off by regulating the spacing between DNA and histones so that you can access chromatin. And anyway, so we published that. So then when I finished my vitreoretinal training at Barnes Retina Institute in St. Louis, came to University of Michigan, and then really based starting my lab 12 years ago on epigenetics and stem cells. So the scientific question is, how do histone and RNA methylation regulate the production of retina from stem cells? And in so doing, being part of the field now for many years, of course, I knew Jeff Stern and Sally Temple in New York. Paul Lee, our earlier chair, kind of started an affiliation with them. And they had a paper right around the time I started as faculty where they were growing post-mortem RPE. They were gathering that from, cadaver eyes, and you could dissociate the cells in single cell culture And some of them, 1 or 2 or 3 some percent like that, would clonally grow, and that would totally cover the plate, and then you could redifferentiate them into, hexagonal RPE, with showing a lot of the functional properties of RPE. Interestingly, these RPE cells, though, they could grow into cartilage, fat, all kinds of cells, which you might even bone, you might encounter in an end-stage Tysical eye, really showing the excitement of how adult RPE stem cells are truly multipotent. That got spun off into a company to use the RPE cells as a source, adult RPE, as a source for transplantation in clinical trials. And that's how I got involved in the current clinical trial that we recently published about, a phase 1/2A trial.
Well, tell us about the potential of stem cells in atrophic AMD.
Yeah, well, in atrophic AMD, you're losing a couple of populations of cells, both the cones and the RPE, and then you're having some degree of remodeling among the existing surviving cells. Oftentimes, the Mueller cells will now tightly attach to Bruch's membrane at the edge and within GA, and that becomes a surgical consideration later. But the idea is that because these cells are not efficiently regenerating the functional RPE layer, of course, we know that RPE can regenerate. Well, what I can say is can migrate. We see that in retinal tears and detachments, but for whatever reason, they don't mount a real regenerative response when RPE cells are dying in atrophic AMD. So the idea is really to can we replace the lost cells from cells that we generate in vitro and then go in by vitrectomy and subretinal injection, and basically replace the cells. Now, the holy grail, of course, is replacing the native function with the cells that you put in. But I think after 12 years of stem cell research in the eye, and obviously in other fields even longer, I think the field has realized that it may not necessarily always be in replacing the function of these polarized RPE or the cell types that you're missing, but perhaps the cells just in those areas can replace on a trophic level some function to help the surviving cells live. So we don't know the true mechanism, but that's the idea. Either that they replace the cells that are lost and do the same function of the cells that are lost, or they're down there and they're like a factory, kind of bathing that area in proteins and other things that may help favor the cells to survive, the host cells to survive, and in so doing either kind of alter the trajectory of AMD, as in could you keep vision longer? Could you reduce the growth of GA or slow it down? And so these are the questions we're looking at as we look really in the phase 1 trial. First, we'll look at safety and tolerability, but we're also looking at secondary points for function.
Well, can you tell us some of the clinical results?
Sure. So in the recently published paper in Cell Stem Cell about a month and a half ago, we showed that we looked at the low dose patients who received these adult RPE, stem cell derived RPE. Those were 6 patients, all of whom that I did surgery on. And this is a dose escalation trial, so there's a low, medium, and there's a lower seeing group, a better seeing group, and there's 3 doses for each. So what really reported is the low dose data, 3 patients who were between 2200 and 2800, and then 3 patients who were between 2070 and 2200. We had 12 months.
They all had geographic atrophy?
They all had GA. And we had 12-month data on the lower seeing group. That's who we start with first, the 2200, 2800, and then 6-month data on the better seeing group, 2070 to 2200. And really what we found on average was about a 21-letter gain at 12 months in the lower seeing group and a 3-letter gain at 6 months in the better seeing group. So our target is 18 patients for this trial. This first paper reported on the first 6. Now that the trial has gotten CIRM funding, it's been expanded also out to Stanford, Ted Lang, and LA, David Liao at LA Retina. And so they've taken on as of this year, they've been activated in California, and they've done, I think, about 1 or 2 patients each. So I think we've reached about 11 or 12 patients for our 18 patient goal. Now, the cells are all manufactured at Cedars-Sinai. That's where Clive Svensson is involved in the trial.
What's the surgical technique like?
Um, it involves vitrectomy. Of course, when the patients come in, they're pre-screened. We can go in more of that later, but the actual surgical procedure is vitrectomy and subretinal injection. Um, and the tricky parts, at least that I see in doing these trials in GA, is it really, because the investigational product is relatively small volume, maybe about 100 microliters or so, it's a very small volume. So compared to many of our colleagues in gene therapy, where the volumes might be 10 times that or 5 times that, I find it more straightforward to know that the cells are in the right place by creating a bleb, a subretinal bleb, um, at the GA or at the edge of GA, and then
Mhm.
injecting the cells into that bleb. So 1 to 200 microliter bleb to open things up, and then go through that same 38 gauge kind of incision um, with the subretinal cannula, then to squirt in the cells. So these are cells that are not on a monolayer. They're not on any kind of scaffold. So we're injecting them through a 38 gauge opening retinotomy. I think that the most challenging thing about uh, dealing with cell therapy in advanced AMD with GA is, as I mentioned before, you have all this remodeling when the cones and RPE are gone. And I've talked about this a lot with Christine Curcio, you have this at histological level, a really tight uh, hugging of the Mueller cells to Brooks membrane. And what that essentially does is there's a very tight adhesion. And when you're making the bleb, it's very hard to predict where the bleb will necessarily go. It's not always that easy to open up the entire area of GA. But our goal is, it's possible that the center of GA may be too far gone. But consistently I can, open up the edges of GA and get about at least 30 to 40% of the GA lesion. But over that takes a little bit of learning of how to do and accepting, initially that those blebs may not go in exactly where you want them to go. So, I would say that's the main part of the surgical technique. Of course, to drive the bleb and the cells in that bleb more toward the GA, I might do a fluid air exchange to use air to push the bleb a little closer. Um, but what we found, of course, the phase 1 trial, so safety tolerability, that we didn't really see any major safety signals. Of course, these patients start with pretty heavy systemic immunosuppression, and they're on that for 6 months after they get the cells. So we haven't really seen really any big immunological response. Haven't seen things like PVR or significant worsening of membranes. Haven't seen retinal detachment. So far, the trial looks promising. Of course, as a phase 1, we're dealing with limited number of patients, so we can just report on, say, the vision improvements we see. We don't know if that, statistically significant at this stage of the trial.
What happens to the OCTs?
Good question. We are comparing the fellow eye, and what we do see is a thickening of the OCT in the injected eye. Now, that's very variable, and I can't say that's statistically significant. There's certainly more variability in the thickness of the OCT. And, we're doing some image analysis to identify, is this, typical change after, just having vitrectomy surgery? There may be a little bit of CME, or there's non-cystoid thickening. But even, after that, we're trying to observe what happens when things settle down the postoperative period. I think it's too early to say, but certainly we see much more variability, more on the thickening side of the eye that's injected compared to the fellow eye. We've looked at the edges, of the GA, again, preliminarily. These cells that we get from Cedars-Sinai, from Luxa, our sponsor, their master cell bank, these are cells that don't pigment. So it's really hard for us to say whether it alters the expansion
Mhm.
of the GA that would otherwise happen. That's definitely happening in the fellow eye. But even if the cells settle in the right place and they get to the edge of the GA or in areas of GA, we would never really see them that well by OCT or by fundus autofluorescence.
Yeah, so you're injecting non-pigmented retinal pigment epithelial cells,
Yeah,
so to
non-pigmented or
speak,
at least poorly pigmented.
poorly pigmented.
Yeah.
What about immunosuppression? These are embryonically derived stem cells, correct?
No, these are they're allogeneic. You're right. So that's the intent for immunosuppression,
Yes.
but they're from I think the master cell bank was derived from a 70-year-old male,
Right.
post-mortem, but it's allogeneic. So it involves mycophenolate, tacrolimus that we're giving the patients approximately 2 weeks or so before the surgery, and they stay on that for the duration, through 6 months. And that's the regimen that they're on. And then, I have one of my colleagues, Dr. Kodati, who's a uveitis specialist, as well as experienced in managing some of these medications, who's part of the clinical trial as well.
Are the immunosuppressive medications well tolerated?
Great question. I think it varies. We're dealing with a population, with GA who generally is 70 years old or above. They may have some comorbid things, that don't necessarily exclude them from a trial, but just based on age and things like that. Um, the tolerance overall has been good, but some of the patients it is hard, and sometimes we've had to shorten up. They maybe couldn't tolerate all 6 months of it, um, but they have done several months of it, and I haven't really, ha ha I've continued to examine them and haven't found that all of a sudden we're seeing a large immune reaction against the graft or anything like that. But I have to say in this area, immune suppression is one of those things we have to keep in mind with this patient population.
What about using autologous transplants, pure pluripotential stem cells, producing them from the cells of the individual in question? Is anybody looking into that?
Yeah, there have been a couple trials, both in Japan, one of the earliest trials in the iPSC world. Um, and then I believe Kapil Bharti's investigational product also involves autologous cells. I think from a industry point of view, generating autologous cells on a per patient level is quite expensive. Number one. Number two, it involves a reprogramming of the cells from an adult state, let's say fibroblasts or hematopoietic cells, white cells in the blood, and then reprogramming them back to an embryonic like state. That involves transcription factors, prolonged culture, stress on the cells. And so at least from the first trial, they did one patient. This was in New England Journal, I think in 2016 reported in. This was in Japan, Masayo Takahashi's group. But they deferred the second patient because they identified at least maybe some signal that the cells might be acquiring changes in tumor suppression genes or oncogenes. And that is something we know that can happen with any prolonged cell culture, number one, but also the reprogramming from somatic cells to embryonic-like stage cells, which is really required for autologous iPSC generation and subsequent transplantation, could introduce, these changes in oncogenes and tumor suppressors.
Well, this is important work because we're interested in the complement inhibition era. We're really trying to stabilize vision, but restoration of vision is what millions of patients are looking for. And in many ways, stem cells are the most promising avenue for offering these patients vision. Would you agree with that?
Yeah, I agree. I think it's stage dependent. I think when you're at an atrophic state and you've totally lost most of your cones in and around the fovea and the RPE, right now I don't really see any other agent besides cell therapy or stem cell-based therapies that can address that, really profound need in late stage disease. I think there's a role for other things at earlier stages, perhaps, but I agree with you. At this stage in the disease, uh, you know, this is probably our most promising candidate.
Well,
Uh,
so what's the next steps in your study? You've expanded it with two more investigators in California.
Right.
And
Yeah.
how many eyes are you trying to enroll?
Yeah. So we've hit somewhere around 11 or 12. So we have 6 more to enroll. And between these 3 centers, we should reach that target. I'm hoping soon. I don't know if it'll be, you know, by the middle of 2026, or I imagine at some point, you know, in the 2026 to 2027 range. Um, and yeah, I think with, you know, now it being a multi-center trial, we should reach that, in my opinion, quite quickly. Whereas, you know, the first 9 patients or so, 8 or 9, were from our single site.
Well, thank you, Raj, for this great discussion about stem cells, and congratulations on your lifelong interest and evolution from the basic science laboratory to the clinic. Good work.
Thanks very much. It's been great to see things go full circle, and we're really excited on what's next.